So muscles, we've already introduced the muscles. So we're just going to be continuing on. You know, up to this point, we've been talking about the big picture. We talked about sometimes more than one muscle is doing the same kind of motion. Sometimes you have the same muscle, like your biceps is doing both like supination and flexion and shoulder flexing, you know, so you can have, we've been talking about the big picture. Another question, like a third of the people missed. What kind of contraction? Generating force, but the muscle isn't changing length. Isometric, same length. What about if I am taking something kind of heavy and like my biceps have to work pretty hard to keep this from just falling down? So my muscles are working, but they're actually getting longer as the chair wins and gravity takes it down. Eccentric, isotonic contraction. If the muscle's pulling, changing length, but it's actually getting longer. And obviously the concentric is if you open the muscle, the force of the muscle wins against whatever's pulling against it. What we're going to talk about now is what actually generates that force. And it's, you know, again, this is stuff you've seen in Bio 110, the old actin and myosin yanking on each other. So we're going to look at that in more detail. We are going to be talking about skeletal muscle today. Obviously, there's cardiac muscle, which we've talked a bit about, smooth muscle, which we've talked a bit about. We'll talk about both of those in a little more detail. The basic actin and myosin pulling on each other is going to be the same in all kind of muscle fibers, but there's details that are different in different kinds of muscle fibers. So everything we're going to talk about right now is going to be focused on skeletal muscles, the voluntary ones, the ones that make up your biceps and your triceps and your lats and your quads and your gluteus and all of that kind of stuff. The muscle cells developmentally usually become this fusion of many, many, many cells. So you end up getting a skeletal muscle cell with this big, huge multinucleate monster thing. Single fibers. In fact, your longest muscle fibers are like sartorius, goes from your hip down to your tibia. Single cell going all the way from the top of your hip down to the lower leg. So then some of these cells are big, huge cells and have hundreds and hundreds of nuclei because, again, developmentally, they're the fusion of lots of different cells. You know, so when I draw a skeletal muscle cell, I'm just going to, like they often call them fibers because there's big, long things. Kind of like with nerve fibers, nerve cells, muscle fibers, muscle cells, just because they're super long and thin. So this is my skeletal muscle cell. A fiber. It goes on in both directions for a long ways. Most things that have to do with muscles are going to have either the prefix myo or the prefix sarco. Myo is from muscle, sarco means flesh, right? If you're most of your flesh, you're eating meat, you're eating muscle. Muscle, moose, what is musculus? Where does this word come from even? What's that? No, it's Latin, it means like little mouse. You know, it's kind of like, hey, look, there's little mice running in my arm or something. So muscle actually comes from the word for little mouse, which is kind of funny. So the cell membrane of a muscle cell gets called the sarcolemma. it is going to have all those voltage gated sodium and potassium channels like we saw in the axon of a neuron. Which means, what is it going to do? If you electrically excite part of this thing, what's going to happen? What's that?
Contract.
No. What does depolarize? You're going to have this chain reaction depolarization action potential across this whole thing. That supports an action potential. That's going to be the first step. Ultimately, it is going to lead to contraction, but through many, many steps. That's going to cause a release of intracellular calcium, which is going to bind a little protein that's blocking binding sites on the actin and all that. So all the sarcolemma is going to do is have this big chain reaction electrical excitation that runs across it. Again, that's what we were measuring in the EKG, because this happens in all the different muscle cells. Part of the contraction process is this big action potential that runs across. Where did we see the actual action potential on the sarcolemmas of skeletal muscle in our EKG lab?
When we were like fists.
Yeah, when you were moving your hands and arms around and you got all that crazy spiking all over the place. Remember that was just the sarcolemmas of your skeletal muscles all having electrical activity swamping out the signal from the EKG because they're closer to your skin there. The sarcolemma, all right. Inside the cell, the actual contractile organelles that are made up of the actin and myosin and some other proteins are going to be the myofibrils. They're striped. Skeletal is also called striated muscle because the contractile proteins are set up in these kind of alternating arrangements, actin, myosin, that make it look stripy. So these are called myofibrils. These are made up of the contractile proteins. And we're going to talk about these in more detail. That, actin myosin as well as focal myosin, troponin and titin, and different proteins in there. The more myofibrils you have in there, the more strength this muscle will ultimately be able to generate. Like when you are working out and your muscles are getting bigger and more defined, you're not getting more skeletal muscle cells. You're having the same number of cells, but you are increasing the number of myofibrils within each cell. So each cell gets fatter in diameter. So we'll talk more about that, particularly like the muscle cells that are optimized for like really strong contractions, the fast twitch white fibers are going to be much larger in diameter because they're going to have way more myofibrils in there. The more myofibrils, the stronger a contraction that cell can do. And we'll look in this in more detail. The other thing we need to look at in this big picture is the sarcoplasmic reticulum. So it's a specialized version of an endoplasmic reticulum. And in the muscle cell, its main job is storing calcium. So let's put in the sarcoplasmic reticulum. it's kind of sitting here on top of everything. This is where I really wish I had there's no way to shade. When you have chalk, you can just put it on its side and you can shade and stuff with pens. You can't really shade, so. Where's calcium? So when the muscle is at rest, there are little ATP powered pumps that are just vacuuming up any calcium out of the cytosol and stores it inside. So we have constantly calcium being vacuumed up out of the cell and stored in the sarcoplasmic reticulum. This calcium is going to be the trigger that's ultimately going to cause muscle contraction when it's released out of the sarcoplasmic reticulum into the cell. So just basically, you're actively vacuuming up the calcium out of the cytoplasm into the, rather the cytosol into the sarcoplasmic reticulum. The one other thing we can add into this picture, the sarcolemma have these little invaginations, basically like kind of dive down into the cell in order to create this. It's actually these little receptors that interact with the sarcoplasmic reticulum. This is called the T-tubule. T-tubule means transverse tubule because there are these little invaginations of cell membrane. This is a sarcolemma. It's kind of going down into the cell. This action potential is going to be what triggers the release of calcium from the sarcoplasmic reticulum into the cell. So, action potential on the sarcolemma. So this is going to be part of this excitation contraction coupling. When the muscle is getting excited, there's going to be an action potential that goes across the whole cell. And that action potential is going to travel down the membrane in these little T-tubules. Is going to trigger the release of calcium from the sarcoplasmic reticulum into the cell. That calcium is going to interact with the proteins in the myofibrils and ultimately cause the contraction to happen. So, does that part make sense?
Can you say that one more time? Sorry. The part, yeah.
So basically, part of the way muscle gets excited, you know, there's actually going to be motor neuron releasing acetylcholine at nicotinic cholinergic receptors on the cell membrane here, which is going to depolarize the cell. Once we depolarize part of the cell membrane, the action potential goes everywhere. As the action potential goes everywhere, that triggers the sarcoplasmic reticulum to release calcium into the cell. And that calcium, in a way we're going to see in a few moments, is going to interact with the proteins in the myofibrils to create the contraction.
And how does the T-tubule factor into that?
It just, so that depolarization that's on the surface of the cell also kind of plunges down so it can interact here. Okay. So now we'll look at the myofibrils and their contractile proteins. There's a few that are important. The first two are the ones that I'm sure you're familiar with, the actin and the myosin. and also called thin filaments. They're little subunits that just assemble into these thin filaments. So in general, I'm just going to draw them as a line, but in reality, they're actually these little subunits that kind of connect up. And they actually, that little groove that forms on the interconnecting units is going to be important. It's going to be where the myosin head can bind onto. Myosin, so another one. Filaments. So myosin is a larger protein. It's actually another thing with quaternary structure. We're going to see three proteins with quaternary structure today. We saw hemoglobin, we saw the multiple globin subunits. Antibodies, which we're going to see in a few moments when we do the blood, are also made up of multiple subunits. And myosin is another one. It's got a couple of heavy chains, a couple of light chains connected together into this thing that has kind of a head and a tail. It's like a single myosin. It has two conformations. It can be either kind of sprung, where the head is kind of gone down, or we can kind of get it into this more cocked conformation. ATP. You can kind of think of this as a, right, and this can, once it's there, it can then spring. And you can like think about on an, I don't know if you, like an old revolver in the Wild West, or if you had a cap gun you used to play with as a kid or something, you pull the little trigger back until it clicks. And then you, not the trigger, you pull the hammer back until it clicks, right? You know, and then you pull the trigger, boom, goes forward and whacks on your cap, right? So it's like that. You use the power of ATP to basically snap it until it's cocked. And it turns out the trigger is binding myosin to actin. Then it's going to spring. So basically what's going to happen is this thing goes, it's cocked, it's got a lot of potential energy stored in just the mechanical structure. As soon as it grabs onto here, that's the trigger, and it pulls, and that's where the force is going to come from. It's a mechanical thing. You literally take this thing, the conformational change, and now it's in this stored mechanical potential energy, and it grabs onto the actin, it triggers it to spring, and in that process, it's going to pull the myosin and actin pull against each other. So that's like at the core of generating the force. The myosins are connected together tail to tail. You've got like hundreds of them. Make a thick filament. So the actual thick filament is more like this. And then the arrangement of the actin and myosin is ultimately what's going to be creating the whole way that the muscle can contract. It's also going to create this stripy appearance. You're going to see actin and myosin are kind of alternating. So let's look at that. So what we're doing right now is we're going kind of submicroscopic down here in the myofibrils. When you look at the myofibrils under a light microscope, you can actually see that it's kind of stripy and it's light and dark and light and dark. That's how the muscle originally got the names of the I bands and A bands and H bands and Z lines and all that. It has to do with the optical properties, isotropic, anisotropic, Helle is bright. So in our class, you don't need to know A band, I band, even though those are still used. Because now we actually know what the actual proteins are that are making up all the different optical bands. So what we're going to do is we're going to just look at a level that you can't actually see in a microscope. So I'm going to be drawing individual molecules that make up the myofibrils. They color coded the thin filaments. Pockets. So this is actin. The actin just forming this system of these little pockets anchored. What are called Z lines. This is the one I will use because the Z lines give us some reference for the actual contraction. Z scission between each of the different little, what we call sarcomeres. One of these little things is called a sarcomere. Sarcoplasmic little single component that makes it up. So little component of flesh. And then we need the myosin, the fixed filaments, just sitting in the little pockets. So ad infinitum, up and down and in and out of the board and into the board. This is just basically the repeating structure of muscle tissue, skeletal muscle tissue. You know, the structural and functional unit of skeletal muscle. So what we're describing here is going to be called the sliding filament theory of muscle contraction. This is a dude named Huxley back in the 50s came up with this originally. The basic idea is when a muscle contracts, the myosin heads are grabbing onto the actin and pull it, right? They're kind of cocked, they reach up, they grab, and the Z lines get pulled closer to each other. And that's where the muscle contracts. It's because the myosin heads can reach up, grab, and then they do that little spring thing and yank and pull. It's called sliding filament theory because the muscle gets shorter because filaments are sliding past each other. Nothing's actually getting shorter except the length of the muscle. It's not like a rubber band where there's an actual elastic thing that stretches and gets shorter. It's like, think about my thumbs are far apart, now they're closer, now they're farther, but my fingers never change length. It's just they get more into the pockets or less in the pockets of each other. Right, so the actin and the myosin don't actually change length. It's the just relationship and how deep in the pockets of the sarcomere the myosin fibers are. Does that make sense? Then obviously we need some way to control it. Remember I talked about if the myosin can grab onto the actin, that's going to trigger the contraction. So if the muscle is relaxed, we need something blocking the myosin from touching the actin. So that is going to be another one of these proteins that's important. Now let's just finish up with this here. Well, one sarcomere. So if the muscle is at rest, there is going to be this other protein called tropomyosin, which sits in, I said there's like that little groove where the actin little subunits go together. The tropomyosin protein goes in, gets in the way. The tropomyosin is blocking our actin from, okay, this is our myosin here. Actin. So the actin and the myosin cannot touch while the muscle is at rest because the tropomyosin is in the way. And obviously we're going to need some way to move it out of the way if we want to contract. That's going to be job of yet another protein in here called troponin. So troponin has a binding site for tropomyosin, for actin, and also for calcium. So if this binds calcium, causes tropomyosin to move out of the way. Basically, what's going to happen is when this thing binds calcium, it has a conformational change. It changes its shape and in such a way that it pulls the tropomyosin out of the way. So if that happens, so tropomyosin is no longer walking. It's called cross stitch. The myosin can now grab on. And then what happens when actin binds the myosin? That's what caused the spring. The myosin springs. Spring. Power stroke. So the myosin springs, and as the myosin springs, it pulls, and that's where we're causing the Z lines to pull closer, the sarcomere to shorten. Does that make sense? What's going to happen next? We had one, this is at rest. Here we have calcium enters. That's moving the tropomyosin out of the way. We form the cross bridge. That's going to allow the power stroke to happen, the actual pulling. What's going to happen next? I should get out of this screen. Gotta let go. Gotta let go. And what else has to happen? What's that?
The tropomyosin protein has to come back.
So the tropomyosin, that's going to depend on whether we want to keep contracting. That it will happen if the muscle's going to go back to rest. And what else? If we've just sprung this thing, what do we need to do? We need to reset it, right? Get it back into the conformation so it can pull again. What molecule has energy to do stuff like that? ATP. So we're going to have ATP now. It's going to be important. ATP does two things. ATP, as John was saying, it actually is necessary for the myosin head to unbind from the actin. And it makes it recoil. And you go again. So this is now back to Easton's thing. If there is still calcium around, it's just going to grab again and keep pulling. If we're done with the contraction, the calcium kind of gets vacuumed back up into the sarcoplasmic reticulum. So then the muscle's just waiting for the next round. So this is at the core of muscle contraction. The sarcoplasmic reticulum is holding on to this calcium when the muscle's at rest. If the muscle's being excited, the calcium is released into the cell. That calcium binds the troponin. The troponin moves the tropomyosin out of the way, which allows the actin and the myosin to bind, which creates the contraction. Then ATP comes in, resets the myosin, and this is also what happens if you're dead.
Rigor mortis.
Rigor mortis. Why do you, has anybody ever seen a person or an animal in rigor mortis? It's intense. Literally like rock solid, like you can't move them at all. And why is that?
There's no more ATP to bind the myosin.
Yeah, so when you first die, there's some ATP left over and some metabolic pathways to keep making some ATP for a while. But eventually you run out of ATP. And at that point, whatever length the muscle is at that moment, it's frozen there, it's locked. The myosin heads can't let go unless there's more ATP. So all your muscles are stuck at whatever length they are, and they can't change length. So it means all your joints are locked in whatever angle that they were in when that moment happened. And that's going to last, until the basic tissues are deteriorating. So after a day or two, things will soften up again, probably. But there's a little while where there's enough ATP to keep this process unbinding happening, but after a while that stops. So that is that. The video that I really liked got pulled for some kind of rights violation. But I'm going to see if there's another one I found. I'll see if it's actually useful. I think seeing this in animation is more useful than me talking about it. So well, the other thing, while this is warming up, what is going to keep the muscle from just like slipping? Like, you're grabbing, pulling, but then you have to let go and recock. How come when I let go and recock, the muscle doesn't just slip? So I think it's still under force. Yeah.
Because there's hundreds of different things acting at once, and some let go, others.
Yeah, exactly. They're not all letting go at the same time. It's like if you're doing tug of war with a big rope, it'd be kind of stupid if you all let go at once and try to regrip, because then it would slip out from under between your hands. So it's like one person repositions and another person, but you don't all do it at the same time. So the same thing with the myosin heads. They're doing it kind of asynchronously. So you're never just letting go of the. Oh, this could be useful for you this weekend. Beautiful, relaxing piano music for stress relief and study. Hip hop beats to study and relax too.
That's the best time.
I wonder if it figured out we're in a school room. That's weird.
Well, it's like really like
Yeah.
it's like super mellow. It's not like actual.
It's
hip-hop.
good. The girls
Oh, you've listened to it before?
Yeah, it's
Yeah.
a good station.
Oh, wow.
Lo-fi is good.
Oh, I didn't even know these existed. All right.
Just like
Yeah, yeah.
That particular one is famous.
leave it.
Yeah, they're super-
One of your-
When lung has slightly flux,
Oh, oh.
each muscle is linked to the number of bundles of muscle fibers. Each bundle of muscle fibers contains anywhere from 10 to 100 fibers. Each muscle fiber itself
Oh, this is tripping.
organelles known as myofibrils, which themselves are bundles of proteins called actin. Surrounding the myofibril, there is a network of tubules and channels called the Sarcoplasmic Reticulum, where calcium is stored. Each myofibril is broken down into functional sections called sarcolemma.